Hi friends, are you excited of planning blue-white screening? Even me too. Come, let us refresh together what is blue-white screening. Imagine you have a plate, right? Contains your recombinant DNA and you are confused whether the recombinant DNA is correctly inserted into your clone or not. To know it, there are several screening techniques are there. One among them is blue-white screening. We all know the plasmid vector is a circular DNA in which consists of three important regions: origin of replication, antibiotic resistant genes, and multiple cloning sites. So, origin of replication, as the name suggests, from where the replication starts. And the antibiotic resistant sequences consist of different antibiotics. Here we will look over ampicillin resistance. And multiple cloning sites are the sites where endonuclease will come and bind. And are unique sites because this site is nowhere exist in the plasmid vector. In that particular example, we are going to see about LAC-Z, where MCS lies between the LAC-Z and the peptide sequence. This LAC-Z will go codes for beta galactosidase enzyme. These enzymes will act upon lactose to break into galactose and glucose. So beta galactosidase acts on lactose to break it into galactose and glucose. Be clear with this point. If we look at the structure of beta galactosidase, it is a homotetramer. As the name suggests, the same unique molecules forms 4 times of 1024 amino acids. So, if you take the hand, the entire hand is not active, right? Only the three fingers are useful for writing. Similarly, the 1024 amino acids, the few regions are required for active site and the few regions are required for structural stability. Here, the 1024 amino acid sequences is divided into alpha peptide and omega peptide. The alpha peptide exists in the internal regions, and these alpha peptides are responsible for structural stability. So how we are going to relate these information to our content blue-white screening? Well, take a E. coli strain. In the E. coli strain, genome consists of LAC-Z deficient, means LAC-Z lacks the alpha peptide. Out of the 1024, the first 40 amino acids are lacking. Here we are going to see about two important chemicals, IPTG and SGAL. IPTG, if I expand, it may fear you. You want to know it. Isopropyl beta D thiogalactopyronoside. So do not fear. It's nothing but allolactose. Another chemical is XGAL. So XGAL is again 5-bromo 4-chloro 3-anodal so and so. Fine. So what is this XGAL? This XGAL is nothing but analog of lactose. So instead of giving lactose exactly, we are giving for XGAL. That is the reason behind it. So this allolactose, that is IPTG, will activate the lactase expression and produce beta galactosidase. Whatever beta galactosidase will do, we have seen it will go and act on lactose, right? But here instead of lactose, we have analog of lactose called XGAL. Whew. Hope you are taking deep breath, friends. Now it's time to make your eyes sharp because we are going to further molecular details of it. Look over the E. coli genome where we are treating with IPTG. So when we treat IPTG on the lac promoter of the E. coli genome, it will synthesize LAC-Z minus, means lack of alpha peptide. Now same IPTG of course will interact on the vector DNA also, right? The circular DNA of bacteria. When it interact over it, it will produce alpha peptide expression. Now the both proteins, which is LAC-Z minus, consists of only omega peptide and alpha peptide. When it interacts, they form alpha complementation. As we have discussed in the beginning of our lecture, that alpha complementation will lead to the formation of homotetramer. So this homotetramer is a stable structure, and this homotetramer will activate beta galactosidase enzyme, which further activates on XGAL. So XGAL is nothing but an alternative form of lactose, okay, an analog of lactose. This XGAL will form dimer further. This dimer is responsible for formation of the blue colors. So if the blue color exists, yeah, you are right. The DNA doesn't integrate it into your clone. Whew, you got that eureka moment. Means your gene of interest is there in your clone vector. Means what? Your lac is at alpha is not produced and there won't be alpha complementation. Of course, there won't be no beta galactosidase active enzyme is produced. Simultaneously, there will be no activity of XGAL. So finally, there will be no blue color.
Oh, understood.
Hope we made you clear and you may understood the screening technique, right? To confirm the clone, if white colony forms, your gene of interest is ligated into a vector. If your blue colony forms, your gene of interest is not ligated into a vector. Fine, that's all. Enjoy cloning.